Hi everybody, my name is Isabel Nazarian and I'm here from Ohio State University with the Department of Entomology to talk a little bit about some research I'm doing to assess the importance of goldenrod as a foraging resource in colony health. First, I wanted to give you guys a little context as to who I am and how I got into all of this. I am currently in my 5th year getting my bachelor's degree at Ohio State University, and I am finishing up studying environmental science and archaeology, hopefully graduating in the spring. My area of knowledge and specialization is actually in ecosystem restoration, which I love being able to apply to a variety of different things that I learn. I previously worked in environmental education for 4 years, first in Licking County and then out in Kodiak, Alaska. And finally, I joined Reed Johnson's lab in February of 2020, where I have primarily been helping out with data collection and tending to all of the beehives. So I first got into beekeeping in 2017 while I was attending Miami University, where they have a beekeeping club. Once I joined Ohio State in 2018, I took Reed Johnson's beekeeping class, and then 2 years later, I joined the research lab that he leads. Previously, I worked on research involving colony mortality rates in response to neonicotinoid exposure after corn planting. Some of my special interests have to do with honeybee and plant relationships and ethnobotany. I hope to continue working on how management decisions in restoration ecology and everything that I've learned thus far can benefit beekeepers. I'm currently studying the relationship between colony strength and goldenrod availability at the end of the foraging season. So to do this, we're focusing on 2 plots of Conservation Reserve Program land out at Farm Science Review in conjunction with 6 colonies that are within foraging distance. With this information, we will be comparing the progression of colony weight and nectar composition in trends with goldenrod bloom, time, and quantity. And this is actually part of a much larger study on honeybee support in the agroecosystem and relationships between resource availability and CRP land being conducted by Harper McMinn Souter, who I work really closely with and collect all of this data with. So as you guys probably know, goldenrod is the cornerstone of late summer and early fall foraging, starting off in early August and often lasting all the way through the end of October. It's often one of the last sources of nectar and honey that the bees can collect. Its large presence creates an opportunity for substantial late foraging in preparation for the winter. This influx of resources stimulates the queen to produce more brood and strengthen populations, more honey for the winter, and an excess of pollen to provide protein. So altogether, this basically creates a perfect recipe to encourage winter survival. Something that I wanted to talk about was that there's just not one, but there are many species of goldenrod in Ohio. Many are really difficult to tell apart, and they often hybridize, making it even more obscure. But realizing the diversity within the goldenrods is a really interesting fact that I didn't learn until recently, and I don't know how many people really realize how many different species there are. So I started off, I just want to show you guys some different photos of different species. These photos show some structural differences between probably the 2 most common species of goldenrod in our CRP plots, Canada goldenrod and gray goldenrod. These 2 are some that are much easier to tell apart: stiff goldenrod, which has these rounded felty leaves, and grass leaf or flat top goldenrod, which its name describes pretty well. And lastly, a few other interesting species are giant goldenrod, which can get to be about 7 feet tall. We actually have our own species of goldenrod called Ohio goldenrod, and showy goldenrod, which probably has the largest flowers and is probably the most ornamental. These 3 are less common to find in the CRP, but I still see them around every once in a while, and they're pretty cool, so I wanted to mention them. Now on to the methodology that I follow to conduct this research. We have 2 plots, each comprised of 10 by 10 meter transects. Each transect is surveyed by walking back and forth through. Every flowering species is identified and recorded as present within that transect. Then we conduct subsects, which are 5 meters by 5 meters, or a quarter of the larger transect. And these are performed every few, where we count the floral units of each species and record them. We try to survey about every 2 weeks. But sometimes that doesn't happen because of weather, but so far we've had pretty good luck. Now for the beekeeping aspect, every time we have a floral survey, we try to get out and get to the beehives too. Each time weight data is downloaded from the brood minders that we have. Colonies are checked for general health, presence of pests, disease, queen status, and we collect open nectar samples. Lastly comes the lab aspect. Back in the lab, we analyze the honey. Nectar samples are diluted, given a benchmark, and pollen is extracted through 2 rounds of centrifuging. The extracted pollen pellets then go through one or both methods of identification. First is microscopy. Every plant basically has pollen that takes a unique shape and can be identified like any other part of a plant. It takes a lot of training, but under the microscope, you can identify them, and they're usually roughly quantified this way. The other option is metabarcoding, which is a lot more intensive. This method sequences all of the DNA present in the pollen sample and gives you a biological barcode that you can then search through a database for matches and identify what plant that pollen came from. So at the time of making this, we're still recording data and we haven't gotten that all together yet, so I'm not really sure what we're going to see, but there are some trends that are definitely predicted to surface. Firstly, I predicted that colony weights will increase as goldenrod bloom increases. This would indicate more resources stored within the hive and a larger population overall, an increase in colony health and size. I also predict that these trends should be mirrored in the nectar samples, and honey analysis should show an increase in goldenrod pollen present in the nectar as the goldenrod foraging increases. So there are some larger implications and decisions that can be made from the data that we end up collecting and the consensus that we come to. Highlighting the importance of goldenrod to honeybees and pollinators can, in general, just help guide management decisions, which is really important. You can spread awareness about the importance of programs like CRP, help farmers understand how these programs can create circular benefits for them, and ensure CRP land has everything that it needs for pollinator success. In a formal sense, those working in restoration ecology can understand the late season importance of goldenrod, avoid any disturbance events while goldenrod is flowering when it's the most useful to pollinators, and find alternative methods to goldenrod control, encouraging, accepting, and embracing its presence on the landscape. Informally, those looking to support pollinators in their yards and on their personal property are also made aware of this relationship. Embrace the support and beauty that goldenrod offers, making it a part of their landscaping or avoiding mowing in areas that they can. And finally, we just want to be able to take some responsibility off the beekeepers. Beekeepers have enough to worry about this late in the season. When landowners and ecologists understand the benefits of goldenrod, they can help beekeepers by supporting a late season food source and helping the bees get that last boost before winter so that hopefully beekeepers can rest a little bit easier knowing their bees have everything they need to succeed. So that's all I wanted to share with you guys today. I'm really thankful that everybody is here listening to what I have to say, and I'm excited to hear from everybody else, and I hope everyone is having a good time. Thank you.